We have just looked at the details of embryo development or embryogenesis in both dicots and monocots. But remember that the zygote is not the only product of double fertilization. We also have the primary endosperm resulting from the fusion of the sperm with the central cell. In this lecture, we will look at the process of endosperm development, which, together with embryo development, leads to seed maturation. Endosperm development is important for the proper development of the embryo. It can occur before or simultaneously with embryogenesis. There are three types of endosperm development depending on the plant species. These three types are the nuclear endosperm development, cellular endosperm development, and helobial endosperm development. In the succeeding slides, we will look at the properties of each type of endosperm development. Let's begin with nuclear endosperm development. With this type of endosperm development, the primary endosperm nucleus divides repeatedly without cell wall formation to produce a large number of free nuclei within the central cell cytoplasm. The central vacuole then pushes the nuclei to the periphery of the cell, as you can see here. The vacuole then decreases in size and ultimately disappears as the nuclei proliferate. Finally, the multinucleate cytoplasm cellularizes to produce a multicellular tissue. In some plants, cellularization is incomplete. A good example of incomplete cellularization can be observed in coconuts. When you open up a coconut, you get the white solid part of the coconut and the coconut water. The water is actually composed of cells that did not cellularize. In both monocots and dicots, the primary endosperm resulting from the fusion of the central cell with the sperm cell undergoes mitosis without cytokinesis to form many nuclei. The illustrations in the top panel depict endosperm development in corn, which is a monocot, while illustrations in the bottom panel show endosperm development in Arabidopsis, which is a dicot. During endosperm development, the central vacuole, abbreviated as CV here and here, pushes the nuclei, which are the black spots in our illustrations, here and also here, towards the periphery of the cell wall. Cellularization is then initiated at the alveolar stage. Cellularization starts at the micropylar end and progresses through the peripheral and chalazial endosperm. This now is a fully cellularized endosperm of corn and of Arabidopsis. Another type of endosperm development is called the cellular endosperm development. In this type, the primary endosperm nucleus divides repeatedly, accompanied by cell wall formation as indicated by this black line here. This type of endosperm development produces an endosperm that is cellular from the beginning. Examples of plants with cellular endosperm development include species under the genus Impatiens, Datura, and Petunia. Last but not least is the helobial type of endosperm development. This type of endosperm development is rare and is actually an intermediate form of both the nuclear and cellular types of endosperm development. During helobial endosperm development, the first division of the primary endosperm nucleus is accompanied by the formation of a horizontal wall, just like what you see here. The cell at the micropylar end of the embryo sac then undergoes multiple rounds of free nuclear divisions, while the nuclei in the cell at the chalazial end may cease to divide or divide only once or twice. In summary, following double fertilization, the zygote and primary endosperm will undergo several rounds of cell division to develop into a mature embryo and endosperm. Endosperm development can be divided into three main stages: cell division, cellularization, and differentiation. Endosperm development can occur before or simultaneously with embryogenesis. Endosperm development can be nuclear, cellular, or helobial. The mature embryo and endosperm, along with the seed coat, will form the basic structures of the seed.